This is question number 2, paper 5090, paper 4, variant 2, October November 2024, and this is regarding the plant nutrition and photosynthesis. Photosynthesis in green plants can be summarized by the equation that carbon dioxide combines with water in a complex process, and under the light could be the sunlight or artificial light, and chlorophyll, it is converted into carbohydrate, and mainly this carbohydrate is glucose. Then glucose is converted into other forms of carbohydrate as well, and oxygen is liberated. So ecosystem not only provide food for the plant, but as well as oxygen for the environment and for itself as well, because plant respire themselves as well. A student used the apparatus shown in figure 2.1 to investigate the rate of photosynthesis in an aquatic plant. So see, this is a green aquatic plant. This is the gas jar. A solution of hydrogen carbonate is there, and we can see the bubbles here, and it was connected to the clamp so that it can be kept in this position, in this erect position, and the end of this syringe is blocked so that there would be no effect of the air or atmosphere. And from here, we are providing light for photosynthesis. And now when the student switched the lamp on, bubbles of oxygen formed and entered the syringe. So actually, initially, it was completely filled up to here. The solution was up to here. Water was up to here. But after some time, when the student switched the lamp on, bubbles of oxygen formed and entered the syringe. You can see in the diagram they have shown the bubbles are entering into the syringe. The student recorded the volume of oxygen produced in 10 minutes. So after 10 minutes, he observed the volume, that how much gas is collected. So actually what happens when gas is collected here, as it was closed, so water comes down. So this is the downward displacement of water. Okay, water is displaced downward. The syringe was then refilled with sodium hydrogen carbonate solution. Then what we did, that after 10 minutes, when you have observed the volume of the gas collected over there, then you again remove the syringe and filled it up to the uh, completely filled it. And the experiment was repeated again, and oxygen was collected again. And definitely we know that this is the oxygen gas. We know oxygen gas is produced during photosynthesis. The student then decided to investigate the effect of different colors of the light. So it was the first experiment when he used the white light. Then he decided to investigate the effect of different colors of the light on the rate of photosynthesis, leaving the same plant in the glass jar so that we can compare the result. That's why we have taken the same plant. A red transparent filter is used. A red transparent filter was wrapped around the glass so that the plant only receive the red light. When you use a filter, then only that light could pass, and remaining color cannot pass through the filter. So only the red color can be passed. The volume of oxygen produced in 10 minutes was measured again. This measurement was then repeated. The red transparent filter was replaced by the green transparent filter and then the blue transparent filter. And we repeat the same experiment with these filter. So remember that we have taken three filters: red, blue, and green. When you are doing your paper, then at examination center, then what we should do, we should underline the important points. Red, green, and blue. For each filter, the volume of oxygen gas produced in 10 minutes was also measured twice. So this is the measurement one, this is the measurement two. Actually, we repeat the experiment to avoid any mistake or to justify our result. So it is uh, better to perform different sets of experiments. When there was no filter, then 1.1 was the measurement, and in measurement two, it was 1.3. So the mean of these two values is 1.2. How? Mean is taken by adding as many values are there. So here we have only two values. So after adding, we divide with the number of values. So 1.1 plus 1.3, it becomes 2.4 divided by 2. So it becomes 1.2. So this is 1.2. And similarly, with red, it was zero point 0.7 and zero point 0.9. And similarly, it is zero point 0.8. And for green, zero point 0.3, zero point 0.3. And again, this is zero point 0.3. Now, see what is the question. Actually, definitely in the question, it was asked to do the second measurement. And how to do? Let's see. First, we will see the question. Now, see, on the next page, it was written that the syringe at the end of the 10 minutes for measurement two, the blue filter, uh, measurement two with the blue filter is shown in figure 2.2. So what is the measurement? We can see here that this is 1 and this is 2. So if from volume from here to here is 1 and here to here it is 2. So how many subdivisions are there? 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. It means total 10 divisions are there. So if there are total 10 divisions, so it is 1.1. Sorry, it would be 0.9 because value increases here. So definitely there could be 0. So now use the subtracting method. Subtract zero point 0.1, so it becomes 0.9, 0.8, 0.7, 0.6, zero point 0.5. This is 0.5 centimeter cube. And definitely it would be 0.4 centimeter cube. So we can put this value into our given table, and there should be, I think I have made a mistake. Let me correct it. Okay. So this was the table, but unfortunately, mistakenly I cut it. Anyway, so what was our measurement? It was 0.4. So what should be the mean? 0.6 plus 0.5 or 0.4 divided by 2. So zero point 0.6, and it becomes 0.6 plus zero point 0.4 is 1.0. 1.0 divided by 2 is 0.5. So it would be 0.5. And now what to do? Record the volume of oxygen produced in measurement 2 that we have measured, and we have calculated the mean. So we have done both of these. If you have done, then if it is correct, then you will get the full marks. And using the mean value, calculate the rate of photosynthesis per minute when no filter was used. So how to do this calculation? See, okay, so we should note that we have to calculate the rate of photosynthesis per minute when no filter was used. So come to the table again. Here in the table, when no filter was used, the mean value was 1.2. So what to do with this value? And it was in 10 minutes. This table is when we have taken this volume in 10 minutes. So in 10 minutes, the volume of oxygen gas collected is 1.2. So rate is the any value divided by time. So here the rate of oxygen liberated is the volume of oxygen gas divided by time. So volume divided by time is the formula, and they have asked the time in minutes, and fortunately we have time in minutes already. So unit is same. So the volume is 1.2 in 10 minutes. So your answer is 0.12 cm cube per minute. Okay. Now come to the part B. What they're asking in part B. See, they are asking about to construct a bar graph. So first we should know what is a bar graph. On the grid to show the mean volume. So in the table we have the mean volume. We will take that from the table, and the gas collected with no filter and with the different colors. So we know we have one is the no filter, then red, red filter, green filter, and the blue filter. Okay, and data is there in the table. We will go to the table again. But before going to the table, we should know what are the main points whenever you are going to draw a bar graph. So remember that these points, number one, in bar graph, you must remember that in bar graph or the bar chart, columns are separated. Separated means not merged. When I will draw, then you will understand this, what I'm going to say. And another important point that you should note that is that the graph that is given for you, at least half or more than half of the area should be used. Half or more than half of the area should be used. And uh, another very important thing is to find variable independent variables. So one is the independent variable that you change by yourself, and dependent variable is that which is which vary according to the changes in independent variable. So what is the dependent variable here and what is the independent variable? So the independent variable here are the no filter, these conditions, red filter, green filter, and blue filter. While the volume of the gas collected is dependent on the color of the light. So this is the dependent variable. And remember that dependent variable is always on the x-axis. So this is the x-axis. So here would be the independent variable. Independent variable. So independent variable is drawn on the x-axis, while the dependent variable on y-axis. It should be on y-axis. Here we will draw the independent variable. Okay, so now see in the table, what are the independent variable? This column is independent variable. So different color filters, these are independent variable. These must be on x-axis. And these mean values are the dependent variable. These are on y-axis. So what to do now? See, these values are there and these values are here. Now see how many divisions are there. If we count, then from here, from this corner, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is a dark line. This dark line is 10. So as it is not very much clear, so I'm using this line to make sure that there is a 10. Okay, as I'm doing here on this software, so I have facility. But even you can do this by marking the point over there. And where is the next marking? Or you can do like this. Don't do like this. You can use this one. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is 10. Then again 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. Then again this is the 10. It is somehow clear. And 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this is 10. Okay. Now, another thing, as I have told you that half or more of more than half of the area should be used, and on axis it should be distributed evenly. For example, if you have to draw the four bars, one is with no color, means no filter was there, and another with red color, red color, and then green, and then blue. The width of the bars must be same. You cannot do the different width. So if you take 2 from this center point to the right, then 2 from left, 2 from right. Again, 2 from left, 2 from right, and use your scale. 2 from left and 2 from right. And again, 2 from left and 2 from right. Okay. But what should be the height of these filters? So now see, when there was no filter, it was 1.1.2 centimeter cube. So what are the variable? We should mention that what we are using here. Here we are using volume of oxygen gas collected. Volume of oxygen collected. And mention the unit as well. The unit is centimeter cube. So this is the dependent variable. And now we have to put the values. First, we will see what is the scale. What should we, what scale we should take? 1, 2, 3, 4, 5, 6, 7, 8, 9, and this is 10. Then again, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Then 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So if we take this whole length as 1 centimeter cube, then it would be difficult to plot 1.2 because this the largest value would be beyond the graph paper. So if we take 1 here, then it would be difficult to distribute this value because these are the 30 small divisions. So better to take one here. This is 1.0 and this is 2.0. If it is the case, then it means each small division, each small division would be zero point 0.1 centimeter cube. So this is the zero point 0.1. So this is 0.2. So two small division is equal to 0.2. No, no. Two small divisions is equal to 0.1 because there are 20 divisions when we have taken 1.0 here. This is not 10, this is 1.0. 1.0. So if it is 0.1, then 0.2, then 1, 2. It is 0.2, 0.3, 0.4. This is 0.5, 0.6. Again, two division, 0.7, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Okay. So यहाँ पे here we can take the our readings. So one is the 1.2. So 1.2 would be 1.1. 1.2 would be here. So there would be 1.2. So on the grid, this is the mark for 1.2. And for red, that was 0.7 and 0.9. It was 0.7 and 0.9. Again, I have disturbed the table. So do it again. During the examination, you will do it on paper. So there would be no problem. So for red, it was 0.7 and 0.9. So the mean was the 0.8. So where is 0.8? This is 0.9. This is 0.8. So this is the line. You can use your scale, put the scale over here, then you can mark the point. And for green, there was zero point 0.3. So where is 0.3? 0.4, and this is 0.4. 0.3 is this. This is 0.3. And after that, for blue, it is 0.5. So this is 0.5. Okay. So after marking, you just draw the bar by using your scale. So right now I can draw like this because I'm using this tool. This is one bar. This is second bar. This is the third bar, and this is the fourth bar. So you can shed them as well, but I'm not going to shed anything for saving my time. So this is the bar graph that they asked to draw in our question, that construct a bar graph. So what I was telling you in first point, that in the bar graph columns are separated, not merged. So what does it mean? It means you cannot start the next bar from here. That was the histogram. In histogram, these bars are merged together, but in bar graph they must be separated. Okay. So remember, whenever you are going to draw the bar, you're going to draw a graph, then you should note this point and remember this point before drawing any graph. Okay, now see, the next part question is saying use the data given in the bar chart to state one conclusion that can be made from the result of this investigation. So whether you will see the mean in the table or the graph, because graph is obtained from these values. So answer would be our answer would be same, and you can see from the bar graph or the value that when there was no filter, the most photosynthesis occurs. That's why more oxygen gas is collected because time was the same. And if we see among the individual colors, then red is more effective. So red is more effective as compared to other two colors. And green light is the least effective. So the main colors that a plant absorb is red and blue. So we can write, there are many conclusions that can be obtained from there, but they have asked to state only one. So we can write that red light red light causes more photosynthesis, or you can write uh, red light is more important for photosynthesis as compared to green and blue. Or you can answer in other words that white light when there is no filter is more effective for photosynthesis as compared to individual colors. So you can write any conclusion. There are several conclusion, and you can see the marking scheme as well. Okay, so this is the last component question of component question. And here they are asking, this is the planning question. It has 6 marks. Plan an investigation that you could do to determine the effect of light intensity. In the experiment that we have seen, there we have used different colors of the light. And here they're asking about the light intensity on the rate of photosynthesis, and definitely light intensity affect the rate of photosynthesis. And we will use the same apparatus that we have seen in figure 2.1. Then what to do? So here we can use different light intensities in both the cases. Whether you perform with not whether we should define our investigation by using the same experiment that we have seen here, that in a gas jar we have taken the aquatic plant and clamp it. We will use the same experiment, just we change the light intensity. So first, how can we change the light intensity? Light intensity can be changed by using different bulb. You can use another bulb of the high greater light intensity, or you can change the distance. If it will be near, then light intensity would be greater. If it will be far, so light intensity would be smaller. So, or you can use a dimmer switch with your bulb, and by that dimmer switch that we use for fans in our houses, so the dimmer switch can change the intensity. But remember that whenever you repeat your experiment, then you should remember that what light intensity you have taken for the experiment with no filter, with red filter, green filter, or blue filter. So we will repeat the same experiment so that it can be comparable. And we will refill refill the syringe again and again. As many times you will repeat the experiment with no filter, with any color filter, in all the cases. So in this way you will write your answer. So these were the important point that you will keep in your mind. Remember that this is the question of the 6 marks. So now I'm writing that answer. Okay, now see, I have written the answer meanwhile, and see, you can use your own words, but you should cover all these points I will mark. We will set up the same apparatus shown in figure 2.1, and figure 2.1, but with the same plant. Your answer must incorporate this, that the method, if you are using, then in all the experiment, the same plant will be used. And how to change the intensity, then you can use any one of them. Not necessary that you incorporate all these three in your answer. You can use uh, any of these methods for keeping the different intensities. So the different intensities, or the 3 to 4 or 5, whatever the intensities you need, you can change either by keeping the bulb at a distance, or change the different bulb of different intensity, or the more easier is to use the dimmer switch. So dimmer can change the intensity. So any one of them is must from this point. Then during the experiment, following variables will be kept same, that is the temperature and the concentration of sodium hydrogen carbonate. So this is also an important point. So 1 mark is for this, 1 mark for any one of these, and 1 mark for these two. No, it both has separate marks. And as per investigation given in figure 2.1, syringe will be refilled or will be filled again and again whenever you are performing the experiment with different intensity. So you must mention this as well. So 1, 2, 3, 4, 5 marks. And the main reason for this, that we will wait for the we will wait for equilibrate the conditions. So up to here we get the 5 marks, 1, 2, 3, 4, 5. And now the moreover, at each light intensity, we will measure volume of the gas produced for the same time duration. So this is another important point. So for such planning questions, uh, be relaxed to answer and to cover all the important points. Okay. You can use your marking scheme as well, that what marking scheme says to you. Okay. So that was the last component of this question.